you also looking for answer to the questions like why are there so many new variants of the virus coming up every other day? Or will there be a third wave? Or will the virus ever go away? If yes, then I would say you have clicked the right video because in this video we are going to talk all about it and answer all your questions. So let us say that this is the virus that we are dealing with. But why is it called so? Severe acute respiratory syndrome coronavirus 2? Why so? Because this virus belongs to the same family of viruses which caused the severe acute respiratory syndrome outbreak in China in 2002. Yes, so this is the second virus from the coronavirus family which has been discovered and which has caused such a huge outbreak. And hence this new strain came to be known as the novel SARS CoV-2 virus. Novel means new. And the term corona is a modified version of a Latin word which means crown, because when scientists first observed this virus under a microscope, then it appeared that the virus is actually wearing a crown because of these spike proteins. Now let us talk a little about the structure of this virus. Actually, most viruses are made up of a protein coat and a genetic material. When the virus is present inside a host cell, then it can replicate its genetic material and form new protein coats and thus form new virus particles. Just like other viruses, this virus also has proteins. This red part is the nucleocapsid protein. This blue part, which is enveloping the nucleocapsid protein, is known as the envelope protein. And these yellow parts, these yellow structures which are coming out from the envelope protein are known as the membrane protein, and the black parts are the spike proteins. Okay, so we have talked about proteins, but there is another part as well, the genetic material of the virus, which is displayed in purple color. Now we know that genetic material are mostly nucleic acids, right? So here also the genetic material is nucleic acid. But is it a DNA just like humans, or is it RNA? Well, in coronavirus we have an RNA genome, and that too a positive sense RNA. Now what does that mean? Okay, so let us understand that. Have you heard about the central dogma? The central dogma says that from the DNA, RNA is formed, and from this RNA, protein is synthesized. And this process by which RNA is formed from DNA is known as transcription. And the process from which protein is synthesized from RNA, that process is known as translation. Now I am going to draw a DNA. You know that DNA is usually double-stranded. So I'm going to draw one strand in red and the other strand in blue. And the two strands of DNA are connected with the help of hydrogen bonds. Here the yellow lines denote the hydrogen bonds that connect the red and the blue strands of our DNA. So this is the DNA. Now after transcription, our RNA will be formed from the DNA. We saw that DNA is double-stranded, but you know what? RNA is actually single-stranded. So in order to form this single-stranded RNA from this double-stranded DNA, we will need only one strand that will act as a template. So let us say in this case, this blue strand is acting as a template. So that means that this RNA will be complementary to the sequence of this strand, this blue strand. Also, this red strand is again complementary to this blue strand. So actually, our RNA will be complementary to the blue strand, and the sequence of this RNA will be same as the sequence of this strand, which is not actually acting as a template. Hence, this is known as the coding strand or the positive sense strand or simply the sense strand. And this strand of the DNA which acts as a template for the synthesis of RNA is known as the non-coding strand or simply the template strand or the negative sense strand. This is the case of normal transcription, but we have also discussed that our coronavirus has a positive sense RNA, which means that a coronavirus has an RNA which is something like this, and it can directly code for proteins. So that is why the coronavirus has an added advantage that it does not have to undergo transcription initially. It can simply undergo translation and form all the proteins that are necessary for its replication and multiplication. So this all explains the structure of our or the basic structure of our SARS CoV-2 virus. But now, in order to understand why new variants of the virus are coming up so quickly, so rapidly, we need to look deeper into the structure of RNA and DNA. Now, three major components are generally present in DNAs or RNAs. Number one, a sugar moiety. Number two, a nitrogenous base. And number three, a phosphate group. So the main difference lies in the sugar moiety. In DNA, we have deoxyribose sugar, while in RNA, we have ribose sugar. This is our deoxyribose sugar which is present in DNA. There are 5 total carbons, and on the third carbon, we have a hydroxyl group. But in the second carbon, we do not have any hydroxyl group. There is a simple hydrogen attached to it. Now coming to our ribose sugar, we see that at the third position, third carbon, we do have an hydroxyl group, and in the second position also we have a hydroxyl group. So there is an extra hydroxyl group in ribose sugar than in deoxyribose sugar. And because of this absence of an extra oxygen in deoxyribose sugar, we call it deoxy. The presence of this extra hydroxyl group in every nucleotide of RNA makes RNA more reactive than DNA, and hence it is more prone to mutations. So when we begin to answer the question why new variants of SARS-CoV-2 are coming up every other day, then the first point that should come to our mind is the genome. It is because of the genome of the virus. As RNA is more prone to mutations than DNA, so the virus is able to mutate faster, and hence new variants of the virus are produced very rapidly. There is another important thing we should note here, that the enzyme which synthesizes our RNA, which is RNA polymerase, it does not have a proofreading activity, which means that it cannot check if a mutation has occurred, while the enzyme which synthesizes DNA, it does have a proofreading activity. It does checks whether a mutation occurred, and this lack of proofreading activity also adds to the ability of RNA to acquire mutations rapidly. Now coming to our second point, the host of the virus, which are obviously we humans. Now have you heard about bird flus? Yes, the bird flus, where hundreds or maybe thousands of chickens are killed which are infected, right? Suppose your area has been infected with bird flu. So all the chicken present in that area are killed, and all their remains are destroyed completely. Similar is the case with plants. If a particular area is infected with a virus or maybe a bacteria, then all the plants of that area are destroyed, and in fact all their remains are destroyed completely. But that cannot be done with humans, right? Because this is the world of humans, and here in this world we follow the ethics which are created for humans and by humans and for the benefit of humans. So the only seemingly possible option is to impose lockdown, and that is what is taking place round the globe. Whenever a healthy individual comes in contact with an infected individual, the virus gets new opportunity to infect a new host. As soon as the virus gets into the new host, it will get multiple opportunities to replicate its genome. And greater the number of replication, the greater are the chances of mutation. So that is another reason the virus is able to produce so many variants so quickly. Now coming to our third and the final point, which is pressure of selection of the fittest. Now we all know that this theory dates back to the time when Darwin went to Galapagos Island for his excursion. There he observed how organisms have adapted themselves in order to survive in the changing environment. Something similar is the case with this virus also. Now, the development of our immunity against the virus is imposing a problem for it. So, that is why it is constantly under pressure to change its genome, to modify itself, so that it can fool our immune system. And hence, this is another reason why the virus is undergoing mutations so rapidly. Also, there are some mutations which are not so good for the virus, and obviously, they will die. But again, there are several mutations which are going to be helpful for the virus, and that is why the viruses which are acquiring those good mutations, the good mutations which are good for the virus, okay, the viruses which are acquiring these good mutations, they are surviving, and hence we are seeing new variants every other day. So I think we are clear with why the new variants of SARS-CoV-2 are coming up every other day. Now moving on to our next question, will there be a third wave? So in order to understand this, we will have to look at our immune system. So as soon as our virus gets into our body, it faces our immune system. Inside the human body, the first immune cell that the virus encounters are large macrophages. These macrophages engulf the virus and takes it inside the body. This process is known as phagocytosis. Once the virus is present inside the body of the macrophage puts in all the efforts to digest the virus until very small remains of the virus are left. These remains of the virus are presented by the macrophage on its surface. This process is called antigen presenting, and the small remains of the virus which are presented on the surface of the macrophage, these are called antigens. But why does macrophage do that? Well, this is to tell all the other cells that a foreign body is inside our body, and in order to alert the cells to take necessary actions. Once the information reaches T cells, it then alert the B cells which start producing antibodies against the particular antigen. So this is how our immune system reacts. Now in order to fool the immune system and get entry into our body, the virus will have to modify itself. And that is what the virus is currently doing. The new Delta variant has come up with a new coat. This new coat helps the virus to be unrecognized by the cells of our immune system. As a result, it is getting entry into our body and it is spreading like crazy because of its high transmissibility. So coming back to our question, will there be a third wave? Yes, there can be a third wave if we are not very careful. So what can be done? Simple. We will have to wear our masks properly. We should not go out unless it is absolutely necessary. And the third and the most important thing, get vaccinated as soon as possible. If we are doing all these things right, then yes, third wave is definitely preventable. And now it's time for the third and the last question. Will the virus ever go away? Well, I don't think the virus will ever go away completely. But as we all get immunized against the current variant of the virus will eventually fail to infect any new healthy host, and thus it will receive lesser and lesser opportunities to replicate its genome, which in turn will reduce the chances of new mutations. That will help us to prevent genetic drift and genetic shift, which means that there will be no new variants of the virus coming up, as the virus will lack its opportunity to replicate and produce newer variations. So we will be safe as long as our body has the power to tackle the existing variants, which our body can easily do. So I hope I have been able to answer all three questions in this video. In the next video, we will talk about the most popular vaccines that are available